outstanding. So, Christine, Christine's on it. Thanks, Christine.
So in my research group, which is still relatively young, started in 2018, we are interested in questions about the ecology and conservation of amphibians and reptiles, and in particular of salamanders. I'll admit I have a bias. And my prior work has really touched on a lot of different areas. So I've used occupancy modeling to look at species occurrence and detection, as well as species interactions. I have papers and studies on natural history and behavior in salamanders. And then for my PhD, I also did some work with population genetics and phylogeography, as well as in systematics. And so one of my prior collaborations was in describing a new species of salamander in the southeastern US, Valentine's dusky salamander. But I can't talk about all of this as much as I would love to. And so I'm just going to focus on two areas, on natural history and on biofluorescence, and try to get you to agree with me about how these kind of tie back to conservation. I always make the argument that natural history provides a really important piece of the puzzle with respect to conserving species diversity and conserving populations. And then I can't help but talk about a new area of research that's really exciting on biofluorescence across the amphibia. So we'll start by looking at natural history. And if I go too fast or if my audio breaks up, if there's any issues, just feel free to interrupt or post in the chat. So as Craig mentioned, I was an instructor at Southeastern Louisiana University. And while I was there, I was able to teach herpetology. And it was really important for me to try and get my students involved in fieldwork and in research from the start of the process in idea conception all the way through to the end in writing up the paper. And again, this was back in 2017. So this paper has just been accepted for publication at Southeastern Naturalist. It took a little bit longer than I originally intended, but that always happens. However, this was a really fun project that I think does a good job at tying natural history to long-term conservation plans. So I'm going to take some time to talk about the reproductive phenology and the larval period of the Western dwarf salamander. Now, if you are familiar with salamanders, you know that this group is rife with cryptic species complexes to the detriment sometimes of kind of our progress. So a cryptic species is a group of evolutionarily distinct lineages that are difficult to differentiate due to morphological similarity. And in this context, we have this recently described cryptic species complex of dwarf salamanders, all species native to the southeastern US. So this is some work that came out from Kenny Ray in 2017. And one of my collaborators, Bruce Means, was also on this paper. And what they did was they took this thing that we used to refer to as a single species, and using both morphological and molecular data, they split it into 5 different taxa. And you can see the range and the range maps for these various taxa here. And when I say these things are cryptic, I really mean it. I am not confident in diagnosing these taxa morphologically, which is why, as you'll learn, we use DNA. But here we've got at the top, we've got 2 Eurecia paluticola, and then down here we've got Eurecia chamberlaini and Eurecia quadridigitata. Small, kind of brownish gold salamanders. Now, as soon as these guys were split, we're left with this massive gap in terms of our understanding of their biology and their natural history. And with this question of what do we actually know about each of these different cryptic taxa? So, you know, we kind of started to dig through the literature and think, all right, well, this paper was published on populations in this region, but is that really about the species that occurs there? Because in some places they overlap in terms of their range. So how much do we actually know? What do we understand about their natural history, right? And so natural history can sometimes be this fuzzy word that students often don't have a good feel for, but it really encompasses the behavior, the phenology or the timing, and the ecology of organisms. And it's incredibly pertinent for conservation action. So by understanding things like the timing of reproduction and the timing of metamorphosis, we can then build models to predict how amphibians might respond to things like climate change or increased drying of their habitats. We also are left with this kind of really interesting system with these dwarf salamanders, because although we've collectively been referring to this group of cryptic taxa as the dwarf salamanders, some publications, including this one by Lamb and Beamer, for the record, that's Trip Lamb, not me. Some publications indicate that this is not a monophyletic group. So the dwarf salamanders are paraphyletic. And in fact, the Western species, which let me click back and point out, the Western species is what we find over here in the kind of southwestern corner of Mississippi, throughout Louisiana, and in eastern Texas. But that Western species, Eurecia paluticola, may in fact be more closely related to Eurecia salamanders that are in central Texas as part of this Edwards Plateau complex that includes salamanders that are neotenic, so that as adults retain larval characteristics. So we also can't necessarily assume that this Western lineage, its natural history, is going to be very similar to those of other dwarf salamanders, because it may not be closely related to them. But then if it is similar, it's this really cool example of convergent evolution in terms of life history traits in the coastal plain. So we had several questions. And again, I'm working with undergraduates and graduate students in the class to shape this project. So we needed to figure out what species did we have locally in Hammond, Louisiana? What was their reproductive phenology? And what did their larval life history look like? Our field site was very close to campus. It was a walkable distance, which was an important part of this, at North Oak Park, which is pictured down here in the left. And the wetlands in this area, so there's a lake that's not natural and a baseball field further south. But up in here, we have a mixture of hardwood pine forest with intermittent wetlands that vary in terms of their drying times. And so as you can see, this really straight channel here, some of these wetlands are actually like canals that have just been kind of left to deteriorate and been colonized by amphibians. So to figure out what species we had, because I was not confident in diagnosing them via morphology, I designed primers to target a fragment of a mitochondrial gene that was used both by Ray and by other authors for the Site B gene. And then we compared our sequences from 2 or 3 individuals from our population with those published from those other papers on GenBank. We also wanted to make sure that we were getting as much of the picture as we could with respect to changes in the population or timing of particular events. And so we used methods to target different life history stages of these salamanders. For the adults, we utilized opportunistic cover object and visual encounter surveys in the late fall and in the Louisiana winter. I can put quotes around winter now because now I've lived in Minnesota where there's a real winter. And then we also opportunistically surveyed for adults during our larval surveys. To find larvae, we tested out a couple of methods. Now, in my prior work, I've had really good luck using what are called leaf litter bags to sample for aquatic amphibians. These are basically just, you take wildlife mesh, you make a little pillowcase out of it, and you stuff it full of leaf litter and you put it in the wetland. They work really well in some systems to get larval amphibians. So we tried that here, but we actually found, I think because we were sampling so early in the season for these salamanders, that the larvae were getting damaged as we removed them from the bags. So we dropped that method, and instead we switched to focusing on dip netting and on what's called the Bootstep Method, which is something we describe and are included in this publication, which is exactly what it sounds like. In this very wonderful mucky habitat, especially as it dries down, you step with your boot and you get deep, and then you pull your boot up and you dip net the hole you left behind. And what's really interesting, this works seriously, is the larval salamanders will actually flow into the hole that's left by your boot. At first, the students thought I was joking, and then I showed them that it actually worked. So that was fun. Anyways, we focused on sampling for larvae during the course, which was during the March through June period. When we caught individuals, we measured them by hand, or we took batch photographs, and then we measured typical standard measurements like snout to vent length in image J, and then we released them. So what did we find? As you can kind of see from the title of the paper, the species we had at our site was the Western Dwarf Salamander, Eurecia paluticola. And we found that males, reproductive males and reproductive females were present at the site throughout those late fall and winter surveys. So you can diagnose species based on external, or sexes based on external characters. We've got cirri, these long projections from the upper lip in males. And then in females, you can see ova, developing ova through the ventral surface of the skin. We also found females that had been inseminated within the past 24 hours. So if you're not familiar with Plethodonics, so these are lungless salamanders, and that's the group into which this dwarf salamander falls. They, like most salamander species, have internal fertilization, but they don't use an intromittent or a copulatory organ to achieve this. Instead, the males put down a sperm packet, a spermatophore, and then the females pick up the sperm packet with their cloaca. And that sperm packet will actually remain visible in the cloacal opening for, depending on the size, 24 to 36 hours. And so that's what you see here. So that little white dot is a sperm cap in the cloaca of this female. So we found inseminated females in November. Now, the latest date at which we found males that were identifiable as males based on their cirri was in mid-February. And then for larva, what we've got here, so in this big table, we've got various histograms from each of our sampling periods. So the date is in the upper right-hand corner. Let me zoom in a little bit. And then what you've got along the x-axis are snout to vent length measurements. And then on the y-axis is the frequency of individuals falling into that size category. And so this is a really common method of documenting the life history of populations. And we've got it color-coded so that larval individuals are in black, and then metamorphosed individuals are in gray. Again, we did release individuals between sampling events, and we did not mark them in any way. So it is possible that we were resampling some of the same individuals. Larvae and recent metamorphs were present at our site from March through June. And larval lengths ranged from pretty small, 8 millimeters snout to vent length, to up to about 20 millimeters. Recent metamorphs were about 17 millimeters in snout to vent length. And so you can see here, to give you some perspective on size, this is a ruler with, unfortunately, inches right behind the salamanders. And then on top, we've got a recent metamorph. So no longer does it have those external gills, it's also lacking its tail fin. And then on bottom, we have a late stage larva. So the larvae really only start to get this lateral pattern once they're getting really close to morphing out. This individual probably would have morphed out a week and a half after this photo. The first metamorph was found in our 29th April sample. And then metamorphs persisted at the wetland. So they didn't morph out and immediately leave that wetland site. In fact, they stayed at the site all the way until it was almost fully dry, and when we had to conclude the study for time reasons in early June. So what can we kind of say? How do we tie this back to that kind of lack of knowledge? So despite the fact that Eurecia paluticola may not be closely related to other species in that dwarf salamander complex, like other salamanders in the coastal plain, like other Plethodonics, this population at least reproduces and begins larval development in the late fall and early winter. So there's similarity there in terms of the timing of these events. And what's also really interesting, and this is kind of where most of my interest lies right now, is that larval period is potentially really short. So potentially as short as 2 to 3 months. So we estimated the larval period by backtracking. Unfortunately, because again, working with students and we're learning, we wound up missing the hatchling stage. So the really small hatchlings, which would probably have been 6 to 7 millimeters in snout to vent length by maybe a month. So again, this is an estimate of the larval period. This is one of my favorite images. This was taken at the end of the study, and you can see the muck and the mud behind it, and you get really high densities of these metamorphs in these kinds of wetlands in the Southeast. So overall, we've got a similarity in terms of the timing or the phenology of reproductive events and what's potentially a really short larval period. So where do we go from here? Obviously, you should all be thinking, well, you've just got one population. Absolutely true. It's kind of all we could handle in that one semester of class. So from here, what really needs to happen and what a motivated master's student or an undergrad could tackle would be to repeat this kind of a study across more populations, and then also to do this kind of work across other species in the complex. We also need more frequent sampling within a season to make sure that we can capture things like ovipositional periods and hatching periods. And then this is the part that I mentioned being really interested in. So multi-year studies would be really useful to help us try to understand the effect of drying on these salamander populations and on the timing of their life history. So in some of my prior work where I built occupancy models for species occurrence in stream salamanders, what I found was that stream drying affected the probability of occupancy for brook salamanders. So those are the salamanders in the genus Eurecia. And so that's kind of summarized here. And our species at the time was not split out, but we expect that Western dwarf salamander, that its likelihood of occurring at a site increases as the site is more likely to dry. And there's different hypotheses for why this might be the case. So there's lots of interesting questions such as, how flexible can populations be in terms of the timing of metamorphosis based on how quickly their site dries? And what might we expect to happen in terms of that timing of metamorphosis in the kind of long-term climate change perspective? And so again, these are all areas where we can go in the future. So that kind of information can be used to fill in gaps, understanding when these amphibians are moving into their breeding habitats, because the adults basically, when they're not in breeding season, they're spread throughout the forest and then they migrate into these ponds. Understanding when they do that helps us to make management decisions like when to best burn. That's a really contentious issue in the Southeast, because oftentimes when we burn in the fall is also when these small salamanders migrate. Again, understanding the timing of metamorphosis can help us figure out when to perform management activities. And so figuring out the pieces of this puzzle will help to build more robust conservation and management plans going forward. So I'm going to try and very quickly talk about this next part, looking at biofluorescence in amphibians. So as Craig mentioned, myself and collaborator and friend Matt Davis at St. Cloud State have recently come out this year with a publication called Salamanders and Other Amphibians Are Aglow with Biofluorescence. And I want to talk to you about that because this is a really captivating area of study for a whole heck of a lot of reasons. Now, biofluorescence and bioluminescence always get confused. So I just want to kind of outline what the difference is. Bioluminescence is when the organism itself or other symbiotes living within the organism are producing light via a chemical reaction. So you're probably most familiar with bioluminescence via things like fireflies or perhaps cephalopods. Bioluminescence does occur in vertebrates. So it's present across radiations of deep sea fishes. Biofluorescence is different. So biofluorescence is when the tissues or the compounds or the minerals in a living organism absorb light from the environment and then emit it at a different wavelength. And that emission at a different wavelength, that's the light that's fluorescing. It's often at a different color. So green fluorescent protein, which was isolated from a cnidarian, that's an example of a biofluorescent protein. But what's interesting is that you do get examples of species that are capable of both bioluminescence and biofluorescence. And in fact, the same jellyfish from which we isolated GFP does both. So down here, we've got it bioluminescing in blue light. And then that blue light actually excites the GFP to result in fluorescence. Now, biofluorescence is really well known from marine systems, and in particular corals. But recent work within the last 10 or so years that my collaborator Matt has participated in has also shown that other marine animals, including vertebrates, are capable of biofluorescence, including chondriichthyan fishes, so cartilaginous fishes, and ray fin fishes. Now often, but not always, often these are fishes that are associated with coral reef habitats. When we move into the kind of terrestrial landscape though, most of our understanding about biofluorescence is focused on invertebrates, on things like arthropods. So most of the public will know that if you shine a black light on a scorpion, you get a different color. That's fluorescence, that's biofluorescence. But within the last couple of years, biofluorescence was identified in certain species of frogs in the family Hylidae. So in 2017, Taboada et al. Came out with this really great study that documented biofluorescence in the polka dot tree frog, as well as in some other tree frogs in Central America. And then there have been some examples of biofluorescence that's bone-based in reptilia. So in particular, this paper from Protzel was one of the first to document the potential for sexual differences in biofluorescent patterns in chameleons. So what you're seeing here are different images of chameleons, including CT scans of the skull. And in chameleons, they have these kind of bony protuberances into the skin. And those bony protuberances vary in terms of their pattern, and they're also biofluorescent. Now, there are also some other studies with vertebrates, but the big thing is that at the time when Matt and I started collaborating and wondering about biofluorescence in amphibians, it was literally only known from a couple of species of tropical tree frogs, from the pumpkin toadlet in the family Brachycephalidae. And then there was a small natural history note published by David Munoz on the potential biofluorescence of a redback salamander. Overall, though, the prevalence of biofluorescence across amphibia and in amphibians in different habitats was really not known. And that's kind of what got us started down this path. So we wanted to understand how widespread biofluorescence was in salamanders and how widespread it might be in other clades of amphibians. We also wanted to know if those patterns varied across taxa, and if so, how. And then we wanted to understand what we could about the evolution potentially of biofluorescence in amphibians. So was it only present in certain lineages? And that was one of the hypotheses presented by Taboada. They suggested that when we were looking for other biofluorescent amphibians, we should really focus on hylids, and we should focus on those anurans that had transparent ventral surfaces and white reflective peritonia, which is the covering of the internal organs. So we wanted to test some of those ideas. So first we had to find the critters. This was a really excellent experience where we got to collaborate with students. So this is Bailey, one of my research students. She just started her master's in my lab, dip netting for ranid tadpoles to look for biofluorescence in larvae. We were able to collaborate with scientists and curators from the Shedd Aquarium and from the Field Museum in Chicago. I was making connections with individuals that were selling amphibians as part of the pet trade, just to get our hands on as much diversity as we could. And then we had to figure out how to both induce fluorescence, if it was likely to occur, as well as how to document it. So to image biofluorescence, again, remember biofluorescence, that light is a result of tissues absorbing light from the environment. So you have to have a light source. That's what we call the excitation light. We tested 2 different wavelength groups for our excitation light, including blue light in the wavelength range of 440 to 460 nanometers, and ultraviolet light in 360 to 380 nanometers. Now, some animals actually have eyes that are adapted to focus specifically on fluorescent wavelengths. So there are some deep sea fishes whose lenses actually act as a filter to filter out any reflected light and only allow fluorescing light to pass through. Our eyes, go figure, as humans, we can't do that. So to visualize biofluorescent patterns, we have to use a filter. And that's what we call the long pass barrier filter. So what this filter does, it's pictured here. What this filter does is prevent any of the reflected light from the original excitation source from passing through it. And we use different filters depending on our excitation source. So if we're looking at blue light, then that long pass barrier filter doesn't let light below 500 nanometers through. So it's only allowing the fluorescing light to pass through. And then when you hear me talk about the emission spectra, the emission wavelengths, that's talking about that fluorescent light that's being emitted back into the environment from the organism's tissues. So initial surveys really did involve us running around in the field with a excitation flashlight from Night Sea and some really awesome looking shades as our long pass filters, just trying to scan and get any organism we could, see what the pattern looked like. And then we would bring animals back into the lab, take some photographs, again with this same kind of setup. It's just instead of our eye being on this side of the filter, it would be the camera. We also wanted to quantify the emissions that were coming off of these animals that were being fluoresced. And so to do that, we used a spectrometer. So we calculated or figured the average peak wavelength of the fluorescing light, as well as the range of the emissions being emitted. So what we found was really amazing. And it literally became this conversation between Matt and myself of, I wonder if that glows. And so what this video is showing is we had our light, our blue light shining on this tiger salamander. Then I turned off the ambient white lights and I put the filter in front of the camera. So again, this is a live animal. And as you saw, those yellow colors that we would see under normal white light fluoresce a brilliant green. In fact, tiger salamanders fluoresce so brightly, you can actually see the light being fluoresced back without the filter. It just doesn't look green because you're also getting blue light that's reflected. We found that across the species where we tested both UV and blue light, that blue light excited fluorescence that was much more intense. So here's the rear end of a Pac-Man frog. At least that's what it's called in the pet trade. This is a Cranwell's frog. And here, top left, is that individual under a blue excitation light, and bottom right is that individual under a UV light. Now, recognizing that different excitation light sources are resulting in different intensities, patterns, and colors is really important because most of the previously published work looking at biofluorescence in vertebrates has focused on ultraviolet light and not on other spectra. And so that is definitely a direction the field is moving in now, is testing a wider variety of spectra for our excitation lights. In total, we surveyed 32 species of amphibians, and we found that every species was capable of fluorescing to some degree, although the colors and patterns varied. So what you can see here are some of the variable patterns across caudates. Here's an eastern tiger salamander in the upper right. This is a dwarf siren in the bottom right. And then an Amphiuma here in the center. We've got some lungless salamanders on the left, and then a Chinese firebelly newt in the middle. So one takeaway point from this variety of species is that we have fluorescence across taxa in very different habitats. So in taxa that are fully aquatic, like the Amphiuma and the dwarf siren, to taxa that are fossorial or terrestrial, like the tiger salamander, to taxa that are semi-aquatic, like these lungless salamanders. We also found that we saw variation in the fluorescent patterns in anurans. So we've got some of that pictured here. One thing that's really interesting about bufonids, about true toads, is that they actually emit more red in combination with the green than what I have seen with other anurians. And that's interesting. If you wonder about that, ask me later. We found that of the species where we were able to test larvae, the larva also fluoresced. And that in some species, there was a difference between the dorsal and the ventral pattern. So like in this gray tree frog here, the ventral surface fluoresces very brightly, but you'll see an image later about how that dorsal surface doesn't fluoresce as brightly. Typically, fluorescence was brightest where there were bold colors and patterns. So here's an alpine newt, and up top, we've got it under white light, dorsal, and then ventral. To get these ventral images, by the way, oftentimes the salamander was against a flat piece of glass that we then flipped upside down. So if they look a little squished, that's why. But these are live animals. You can also test for fluorescence on recently dead or frozen animals, but we have found that the longer that something is frozen or the longer that it's been dead, you do start to see some change in the degree that it's fluorescing, but that's not something we've quantified. Anyways, that ventral surface fluoresces very brightly. The dorsal surface also fluoresces in this group, but not quite as brightly. What's really interesting too, though, is that areas that lacked bright colors or patterns fluoresced. So on the left here, we have a marbled salamander. I don't have a good photo of these guys, their ventral surface under white light, but what it looks like is just drab gray. But when they fluoresce, basically anywhere that lacked that dark melanin, those darker pigments, especially the cloacal opening, fluoresces really brightly. And I'm not sure if you're going to be able to get the resolution, but you can also see bones fluorescing through the toes of smaller amphibians. We also found cloacal associated fluorescence in the single species of Caecilian we were able to test, right? So if you're not an amphibian nut, we've got 3 groups of amphibians: salamanders, frogs, and then the tropical Caecilians. We were able to test one species of Caecilian, Typhlonectes natans, that we found in the pet trade. This is a species where they are sexually dimorphic in terms of their cloacal anatomy. So this is actually the tail end of a male, and males have these cloacal discs. And this was a really interesting experience in the lab because we, as you can imagine, live amphibians aren't necessarily always the most cooperative when you're trying to get photographs. The Caecilian had no interest in being photographed. And in general, most of its body was not fluorescing much. You can see here, it was pretty dark. But then all of a sudden it twirled around in the container and we saw that cloacal disc, and it was literally like a beam of light. It was a flashlight. It was so bright. We again saw bony fluorescence through the skin and tissues of small individuals. So this is a larval gray tree frog. Urine fluoresces, which is not that surprising because we do know of minerals or ions in urine that fluoresce in other vertebrates. And then interestingly, mucus also fluoresced. So mucus that came off of the Caecilian as well as off the Amphiuma and off of some of the other salamanders would fluoresce a bright green. In general, the peak wavelengths fluoresced across our species fell within that green spectrum when we put them under blue excitation lights. And again, that's where most of our emphasis for this study focused, was on fluorescence in response to blue light. You can also see, though, that that intensity is highly variable among taxa. So to orient you in right here in blue, the Ambystoma has one of the higher intensities, whereas things like that fully aquatic Amphiuma is a really low intensity fluorescence, although it still does fluoresce. We also didn't find any pattern in terms of whether or not certain clades of salamanders were capable of fluorescence. So we found fluorescence across all the taxa we tested. We were not able to test 2 families of salamanders. So we missed the Asiatic salamanders, Hynobiidae, and we didn't test the torrent salamanders, which is a family that's endemic to the west coast of the US. But in general, All of the 8 other families of salamanders were fluorescing to some degree. And then we also found fluorescence in caecilians and frogs. Now, I'm going to talk about this in a second. It's very likely that fluorescence is caused by different mechanisms in these different groups. So this kind of widespread fluorescence could be an indication of an early ancestor of amphibians being capable of fluorescence, and then that mechanism perhaps diversifying throughout evolutionary history. Or we could have instances of fluorescence that have been independently evolved in particular lineages. At this point, we need more information before we can really get into the nitty-gritty of that story. So, big pictures. Biofluorescence in response to either UV or blue light is more prevalent across amphibians than we previously knew. And again, we suspect that there's more than one mechanism. So we did see that pigments in areas with bright colors were fluorescing. Some of the pigments present in the chromatophore units in amphibians include things like pterins and carotenoids and the reflective guanines. These have all independently in chemical studies been shown to fluoresce. And so maybe that's what's responsible for fluorescence in those areas of bright pattern. But then what about areas that lack those bright, bold patterns and colors? Why are they fluorescing? Well, amphibians in their skin do have a really thin layer of that pterin, and that could be responsible for fluorescence in areas that lack bright color and pattern. And so that's something shown here from the Tabawada paper. Again, they're using light that's closer to the UV spectrum, but they did show that that thin layer of pigment does fluoresce in a species of tropical tree frog. So that could be contributing to fluorescence in places like the cloaca or the ventral surface of kind of the more drab amphibians. It's also possible that compounds or molecules in the mucus or in the lymph of these amphibians are responsible for fluorescence. So Tabawada did take their study into histology and isolated what they then named hyloins in the lymph and the glandular secretions of the tree frog, and found that those were responsible for fluorescence in that species. So maybe that's what's causing fluorescence in the mucus of things like the caecilian and the amphima. If you do get a chance, I encourage you to read that Tabawada paper because they did find that the mucus patterns left behind from frogs jumping fluoresced. And so it's kind of like fluorescent footprints being left behind as an animal moves. So now on to the questions that we get, that we should rightly get, and that I and Matt and I both had as we were working on this study. Can they see it? Amphibians, can they see this fluorescent light? Can other organisms in their environment see that fluorescent light? The bigger answer is maybe, probably. So perception, right? The ability of an organism to detect a signal and then to decode any kind of information in that signal. Perception and photoreceptors in the eyes of frogs are able to see green light in the same range of wavelengths as the light that we documented fluorescing. We also know that for biofluorescence to occur, the animal needs to be active under the kind of light environment that can result in fluorescence. And we do know that amphibians are active under conditions that could produce biofluorescence. So they are active during periods where they're out either during the day and sunlight, and they could get UV light, or at night you can get both UV light and blue light during the kind of twilight hours and early dark. So we do suspect that amphibians can see these patterns, or at least some species can. We also know that some of the other species in these communities, like other vertebrates like birds, are capable of perceiving both UV light and fluorescent light. And so predators may be using these biofluorescent colors and patterns to help potentially find prey. Another question we get are, well, why? Why might they fluoresce? And again, this is another really interesting area that can produce a lot of testable hypotheses. And that's part of the big take-home point of this part of the talk is, we've got so many ways to go with this and so many cool hypotheses and questions to tackle. So one of the potential hypotheses put forth by Tabawada et al. Is that perhaps biofluorescence in, particularly in like arboreal tree frogs that are highly visual predators. You can see these really big eyes in this fluorescing polka dot tree frog. Perhaps they're using biofluorescence to help with their visual acuity, to help to be able to see other conspecifics in those low light environments. It could be that biofluorescence is playing a role in communication. So this is a particular area of inquiry that my lab is pursuing right now with grad students and going into the future. What we've got here is, again, that Chinese fire-bellied newt. And newts in the family Salamanderdae are well known for having these really bright warning colors, aposematic colors on their ventral surface to warn predators that they're toxic. And they oftentimes too will perform behaviors to show off those colors. And it could be that perhaps biofluorescence is playing a role in that kind of aposematic signaling system. Or you could have it being used in communication within the species, perhaps during sexual selection. Another idea is that actually it could be used as camouflage. So one hypothesis for why many of the coral reef inhabiting fishes fluoresce is so that they can blend in with those corals on which they live. And so you can see that here. So this is from one of Matt's prior papers where you have a red fluorescing fish happily on a background of red coral and a green fluorescing fish adjacent to green coral. And then, as I mentioned before, this is a gray tree frog. So this is from some work done by one of my master's students in the past summer, where we have green fluorescence that at least is similar to the background on which that amphibian sits. So if the fluorescent color and pattern produced by an amphibian matches its background, then perhaps fluorescence might be used in terms of camouflage. Again, this is all stuff that we have left to test. How might we use biofluorescence? One interesting idea that's gotten a lot of attention from the herp community is whether or not we can use biofluorescence to find cryptic species in terms of species that are hard to pick out from their environments, especially if, say, you're working with individuals who don't have as much field experience. And I think that the answer to that question is probably yes. So what we've got here is a green fluorescing small species of tree frog. This is a Pseudacris crucifer, so a spring peeper, sitting atop a leaf. Under blue excitation light, anything with chlorophyll tends to fluoresce a very bright red. And as I mentioned before, under that same excitation light, most amphibians fluoresce green. So it is possible that perhaps we could use these flashlights to do nocturnal surveys for small-bodied leaf litter inhabiting frogs and salamanders that may otherwise have low detection probabilities just because they're difficult to spot in complex microenvironments. And then there is a whole lot of other work. So I expect that we're about to see kind of an explosion of publications looking at biofluorescence across radiations of tetrapods. We recently found a non-native species of gecko in central Minnesota. It's in the genus Hemidactylus, but I haven't keyed out to species yet. And for fun, we look to see if it fluoresced, and it does fluoresce both in terms of response to blue light and in terms of response to ultraviolet light. And again, we can talk about reptiles later, but there's lots of areas for potential collaboration and exploration. So I know I'm running low on time and I really want to get to questions. There have been a lot of people that have helped with both of these projects. We've received a lot of support from students and from family members, as well as from state and federal agencies. And if there are any questions, I would love to take them now, or you can shoot me an email later. Thank you.
Thanks, Jen. Folks can unmute themselves or put their question in the chat.
So you mentioned that the toad reflected a lot of red light.
Yeah.
So do you
want
have?
to know why that's exciting?
Yeah.
Okay. Yeah. So again, there haven't been wide surveys across toad species, at least in Bufonidae, for biofluorescence. But why I think that's exciting is because toads also are one of the groups that tend to be more active under daylight conditions, or more active in areas of green vegetation. So in things like prairie grasses, and those would fluoresce red as well. So I'm kind of curious to see if perhaps you see a relationship between animals that either fluoresce red to blend in with vegetation, or potentially that fluoresce red to warn of things like toxins.
I see a question from Ned Doctorman.
Yeah, first, thanks for a great talk. That was really interesting.
Thank you.
On the biofluorescent part, yeah, I'm sure you're aware, but there was a paper that came out early 2019 about biofluorescence in flying squirrels.
Yeah.
And I mean, it's just when the person presented that at a meeting, the immediate response by a group of mammalogists was just, we need to go put black lights on everything.
Yes.
And see what happens.
Yeah.
And so, I mean, would you, so you on your phylogeny, the two groups where you didn't find it were ones where you couldn't sample. And so would you suggest that biofluorescence is just common across taxa? And are there other sorts of sensory modalities or ranges that, you know, we're missing just because of our own biases?
Oh, heck yeah. So I do expect that, I expect biofluorescence to be widespread across amphibia. However, I don't expect it to be equal. So I do think that there may have been evolutionary pressures to reduce the intensity of biofluorescence in some groups, which is why part of that difference in terms of it being darker, less fluorescent dorsally, more fluorescent ventrally is interesting. So my mind spins with questions like, could fluorescence have played a role in the evolution of that kind of split color pattern? So yes, I do expect fluorescence across the radiation, but I do expect it to vary, and we've seen that. So right now, you know, I've got a student that as part of his thesis is looking at fluorescence in larval amphibians, and we're seeing some really interesting patterns there. Other questions?
Were you done, Ned? Okay, so I've been thinking about ways of blocking the fluorescence so you could experiment with it. Mm-hmm. And I'm wondering about possibly putting animals in chambers with windows that you can put filters on, and then put others on the other side and try it with and without fluorescence. You know, see how they respond, if they respond,
yeah,
etc. Have there been any experiments or attempts to do anything like that?
Only one that I know of, and I'm forgetting the author. It was a species of, I think it was a species of tropical toad, but they basically built a little. I don't think it was plexiglass, but they built a container that did just that, that filtered out UV light. And then they basically put a live toad in that, and then one in just a regular plexiglass or whatnot container, and they looked for differences in behavior. And they, I believe, concluded that there were differences in terms of approach behaviors, but I need to reconsult that paper. But if you're interested, because that's an area to figure out how to actually do these kinds of behavioral assays. You know, we can do a good bit of speculating by documenting variation, right? Like if we can find that taxa fluoresce right at the same peak as the backgrounds they tend to spend most of their time on, well, that gives a little bit of credence to this idea of camouflage, but we need to be able to test that. And so some ideas that I have are being able to build models where you can actually manipulate the color and maybe control more of the fluorescent pattern that's coming off of it. But yeah, your idea too of basically building a box that dictates what light is coming in and out is another good one.
So is,
Jen,
is there a seasonal, oh, go ahead, Ned.
I was just asking if you could repeat the author's name for the paper that you're citing.
I'm going to look this up and then
Okay.
I will email Craig. So off the top of my head, don't remember, but I have the paper and we'll find it.
Excellent. I did find out a paper you referred to. That was a PNAS 2017. The Tabuada paper.
Yeah.
Okay. So do you expect a seasonal pattern, right? You know, we have short days coming up, and so will you see that the degree of biofluorescence changes seasonally?
A couple of ideas there. So one idea that's really exciting to me, coming from the South, where we have these stupidly long field seasons and really long days, up to the North, where you got to cram that in to a short period of time, is that I see differences in terms of the behaviors of the amphibian populations for species that occur up here, as well as either similar or the same species down south. So like big choruses of chorus frogs, genus Pseudacris, calling just throughout the day up here. Whereas in the South, in Mississippi at least, I'm more used to those not being so active during the middle of the day. And so I'm curious if perhaps biofluorescence may play different roles in those different regions based on light availability. I do think that there's going to be variation that's associated either with environmental conditions or with physiological conditions of the animals. If you've worked with amphibians, you know that some species are capable of color change very rapidly. And oftentimes you catch them when they're maybe cold and they're darker in color. If you test them for fluorescence right then, the intensity is lower. If you hold on to them, let them lighten up, the intensity increases. Again, this is anecdotal. I don't have the data, but I just have the memory. So I would expect to see some variation in terms of things like, thermal habitat, previous activity, things like that.
You think the animals might suppress it too? I've noticed when I sacrifice fish, sometimes the males just as soon as they're on the way out, their colorful patterns just light up really rapidly. And I wonder if sometimes those are suppressed as well, like the fluorescence would likewise be suppressed unless they needed it, right?
I think that in those cases where, right, we've got species or individuals that are in that kind of darker phase and the fluorescence is not as intense, and then they lighten up, so they constrict those cells containing melanin to show more of the background color. I think that's a situation where you've got things like either compounds in the mucus or pigment cells that are responsible for fluorescence. And I'm not sure how variable those might be in terms of what you're talking about, like a startle or something like that. But, and amphibians aren't quite like squid in how majestically they can change their chromatophores. But, if they are able to kind of contract quickly and show off those colors within 30 or so minutes, that might be affecting a signal too, if it's a signal. It's also totally a valid hypothesis that none of this matters, that it doesn't play a role in evolution, but I'm suspicious of that just because of how variable the patterns are. And because I think, as Ned had mentioned before, we're human, our eyes are kind of lame. And so the questions we ask and how we interpret data are oftentimes biased by our own experience. And so we have to be really careful about that. Another question I often get, and then I'll stop my soapbox, is, well, you have to shine a really bright light on them. Yeah, that's true. But, we're also talking about capturing biofluorescence for our cameras and for our eyes. The eyes of other animals are much more, or they can be more perceptive or more sensitive than ours. What's great is that right now we do have labs that are really diving into amphibian vision. So I think Jovanovic is one of the last names of an author whose work we cite. And so we are learning more, but we still have a lot to understand about those systems.
All right. Well, thank you, Jen. I'll clap for you, and anybody who's unmuted can join me. And
thank you guys for coming.
I think a few of us, the folks that are interested in amphibians might want to stick around and chat with you, and anybody else, if you have a few minutes, I have a few questions for you too. So
Yeah, I could probably stick around until about 1:10, and then I've got another meeting.
okay, all right. So is this how expensive would it be to buy the equipment if I wanted to use this for a techniques class?
It's not as expensive as I thought. So another thing I can do when I send the paper over is send you a link to Nightsea. So Nightsea is a company